Hello, and I've had a few requests just to run over the role of RNA at AS level. So here's our learning outcome. Outline the roles of DNA and RNA in living organisms. The concept of protein synthesis must be considered in outline only. That's a little bit unclear as to exactly how far we take it, but you'll see how far we go in this video. In biology, we consider information to flow in a particular direction. It's stored on DNA. DNA stores that genetic information, and that is stored as the sequence of its bases, those A's, G's, C's, and T's. That is a store of information, that sequence of them. Very important word, that sequence. That is the genotype. You'll have heard the word genotype from genetics at GCSE and from all sorts of things. So that is the genotype, the sequence of the bases on DNA. Now that determines the base sequence on M, or messenger RNA, and we'll look at how it does that towards the end of this video. That sequence of bases on mRNA determines, via tRNA, the sequence of amino acids in proteins. And therefore we get this tertiary and quaternary structure, because our amino acid sequence, which is the primary structure of a protein, then gives it its tertiary, its secondary, tertiary, and quaternary structures. The tertiary and quaternary structure give the protein its function, and that is phenotype. So that's how we get from genotype to phenotype, from the sequence of bases on DNA to the structure and shape of a protein, and hence its function, the phenotype. There are three types of RNA. All of them are copied from DNA. We read DNA to make them. RNA is all single-stranded, is not a double helix, and there are three types, as we've said. So it's messenger RNA, or mRNA, and its role is to carry the genetic code from the nucleus to the ribosome, or strictly the genetic information from the nucleus to the ribosome. So it is just a single-stranded molecule of a certain length, depending on the length of the polypeptide for which it's coding. In a eukaryotic cell, that messenger RNA will have to leave the nucleus. It'll go out through a nuclear pore, and then it will be read outside of the nucleus in the cytoplasm. If it's a prokaryotic cell, then messenger RNA has no nucleus to leave, so it just is there right next to the DNA. Transfer RNA carries a specific amino acid to the ribosome. If you're looking to memorize functions of RNAs, this is one definition to memorize. This is what they look like. They are clover-leafed in general shape, and it is, again, single-stranded. So this is just one strand going along here, although what you'll see is there are little bits of binding like this. So we've got this single strand going along here, but this region of it here is complementary to this region here, such that you've got an A here pairing up with a U here and a G here with a C up here. So it does actually give a little bit of a loop, because we have these structures here. Now here we have what is called the anticodon loop, which I can't underline properly, and we have three bases unpaired pointing out in this direction. That's going to be very important when we come on to think about protein synthesis. Sometimes they have this extra loop, sometimes they don't, doesn't really matter. But the other key point is this: they carry a specific amino acid. Each tRNA carries a specific amino acid, and this amino acid here corresponds to the base sequence down here on the anticodon loop. And this sequence of bases here will always correspond to the same amino acid up here. That's reliable. Ribosomal RNA is a big, long, ugly, maybe beautiful molecule. And the role of ribosomal RNA is to form the ribosomes with proteins. A ribosome is a complex of rRNA and protein. rRNA looks like this. again, you can see it's single-stranded. There's no way on earth you're going to be required to remember this structure at AS level, but just see that it is actually single-stranded, even if it does have these pairing regions along it like that. This is a great overview of the role of DNA and RNA. This is DNA. This is meant to represent our double helix. It's double-stranded. We've got a coding strand and a template strand there. The coding strand here carries the code. The template strand is the strand which is used as a template. No surprises there. And the first thing that happens is RNA is transcribed. Let's write that down here. RNA is transcribed. To transcribe means just to write across from one alphabet to another, and that's what we're doing from DNA to RNA. We're changing the base sequence language from A, G, C, and T on DNA, A, G, C, T, to U, G, and A and C on RNA. What will happen in transcription is you have an enzyme here which is called RNA polymerase. Strictly speaking, it's called DNA-dependent RNA polymerase. It reads DNA and makes RNA. And RNA polymerase will be traveling along this strand here, traveling on the template strand. As it travels along, RNA nucleotides, like this one here, this has got a G on it, will bind in by complementary base pairing. Remember that phrase. G binds onto C here. That will have 3 hydrogen bonds between the G and the C. And this molecule here is GTP, guanosine triphosphate. These are triphosphate nucleotides, triphosphate RNA nucleotides. On they come. And then energy from the triphosphate is used to make these bonds between them. 2 phosphate groups are released, and we reform what are called the phosphodiester bonds. Those phosphodiester bonds are between the nucleotides, and they hold them together and give them a strong sugar phosphate backbone, very similar to that of DNA. So we've made our single strand of RNA, and let's say for the sake of argument in this case it is this one here. It is messenger RNA. Let's see what this is. This is a sequence which is complementary to that of the template strand, and it is the same, bar A swapping with U, as the coding strand. So we have the DNA code now transcribed into RNA form. This is very important because DNA is going to be a long-term store of information. It's a very chemically stable molecule and can last for a very long time. It's chemically inert, which is very useful if you want to store your genetic information reliably. RNA is a kind of go-to useful molecule. We make little copies of the DNA out of RNA. We use that RNA for a bit, then it degrades. It's got a half-life of maybe about 20 minutes, so we quickly use a bit of our genetic information. So we turn on a particular gene, and then when we switch it off again to stop making that mRNA, the mRNA breaks down, and we're no longer having that gene switched on. So this mRNA, what happens now is that this ribosome here binds onto the mRNA, and our ribosome chugs along in this direction. And what happens as it chugs along, a tRNA here, whoa, I'm going to try and draw over it. A tRNA comes in and binds to the mRNA. Now the triplet base here will correspond to 1, 2, 3 bases here. And so the sequence of bases here will determine which tRNA comes in to bind. More of that on the other videos. Have a look at protein synthesis for A2 biology if you want an expansion on that. Each tRNA carries an amino acid, and so it brings an amino acid to the ribosome. You'll see by the time we get to this one here that it's got a number of amino acids attached. Well, that's because each tRNA in sequence has brought along an amino acid. So this one comes along here. I'm going to get rid of this arrow. Here is our next tRNA. Now we form a peptide bond between these amino acids, and that elongates the chain. Then this tRNA, that shoots off, leaving its amino acid behind, and so our chain of amino acids increases. So what is the role of RNA in living organisms? Well, it is, in the case of mRNA, to carry genetic information from the DNA to the ribosome. The role of tRNA is to carry the correct amino acid to the ribosome. The role of rRNA, ribosomal RNA, is to form a chemically active part of the ribosome. So a little set of questions to help you just make sure you understand what's going on, and so you can rattle a few things off. Transcription, that is the word for RNA synthesis. It's very similar to DNA synthesis, but can you give 5 differences between RNA synthesis, transcription, and DNA synthesis? And in just a moment I'll flash the answers up, so pause the video and have a go for yourself. Okay, spoiler alert coming up, here are the answers. It uses RNA polymerase instead of DNA polymerase. It uses RNA bases. They are A, U, G, and C. U will bind to A. Will only form a single strand when forming RNA rather than DNA. And only the coding strand of DNA will be read. Whereas in DNA synthesis, both strands are used as templates in semi-conservative replication. Well, I hope that's helped. Thank you.